The authors of this paper wanted to explore new methods for screening yeast display libraries, and so they designed a magnetized yeast cell, co-expressing a target and SSO Phe2, which is an iron oxide binding protein, increasing the specificity of the interactions between iron oxide and the yeast cell. Using this method, they are able to isolate positive clones using a magnetic surface, and then expand those clones in the media to do further mutagenesis and further downstream characterize those individual clones. The authors were able to successfully co-express the target and iron oxide binding protein. Once they were able to do this, they wanted to examine various various proteins that were expressed on yeast cells that were magnetized, proteins that had known binder proteins. An important thing that they found was when comparing to a control cell in 10 to the 5-fold excess, they were able to get a 100-fold enrichment at 20%, about 20% recovery. And they found that they, with only one round of enrichment, were able to isolate very low nanomolar binders. Importantly, they wanted to examine for nonspecific binding use using this method. The construct that was chosen contained an F2A ribosomal skipping peptide, which when cleaved can reduce the hindrance of binding caused by its presence at the C-terminal end attached to the CMYC. They also examined SSO7D, C1GY, which is an irrelevant mutant, but has really similar residue composition at the binding interface. Using both of these controls and others, they were able to see that they could isolate very specific clones despite similarities between the relevant mutant or a presence of the F2A ribosomal skipping peptide, which would show that the full construct is present. Importantly as well, when looking at this irrelevant mutant and comparing it to SSO7D mutants that could bind, those mutants only needed a 2 micromolar concentration to bind to TOM22, but the irrelevant mutant was not able to be screened at these concentrations. When examining this use in isolating mitochondria, they were able to isolate mitochondria much more specifically than endoplasmic reticulum proteins. And this was very similar, very comparable to commercial kits that can do the same. But with their method, they could provide a greater recovery at weak affinities of about 2,000 nanomolars.